Hey guys, welcome back. In this session, we are going to learn about transgenic animals. Before getting into the topic, we must be aware of the term transgene. So, what's a transgene? A transgene can be defined as a foreign gene or gene of interest which is artificially introduced into the genome of another organism, and the entire process is called as transgenesis. With the advent of recombinant DNA technology, the production of transgenic animals is made possible today. Transgenic animals are those whose DNA are manipulated in such a way that they carry a foreign gene or gene of interest in an inheritable manner. What's the need for a transgenic animal? Well, a transgenic animal can be used as a model for studying human diseases, can be used as a bioreactor for the production of therapeutic drugs, can be produced for a larger yield of meat, or simply for ornamental purposes. There are three methodologies used for the production of transgenic animals: by DNA microinjection, by retrovirus mediated gene transfer, by engineering embryonic stem cells. Let us see the first method, that is DNA microinjection. In this method, eggs are obtained from the superovulated female donor. The transgene is introduced into the male pronucleus, which is usually larger than the female pronucleus, with the help of a micropipette. The so-called manipulated egg is implanted into a foster mother, which gives birth to transgenic pups. The second one is retroviral vector method. In this method, the gene of interest or the transgene is incorporated into the genome of the retrovirus. The eggs, which are in 8-cell stage, are chosen for this method and are infected with the recombinant retroviruses. These manipulated eggs are then implanted into the foster mother, which will give birth to transgenic pups. The last method is by engineering embryonic stem cells. In this method, the inner cell mass of an egg, which is in blastocyst stage, are evacuated from the egg first. The transgene is then introduced into the inner cell mass or the so-called embryonic stem cells by transfection. The manipulated embryonic stem cells are grown in an enrichment medium. The grown-up embryonic stem cells are then micro-injected into the blastocyst. The blastocyst is then implanted into the foster mother, which later on will give birth to transgenic pups. To sum up, transgenic animals are animals which host a foreign gene. Transgenic animals can be produced by DNA microinjection, retrovirus mediated gene transfer, and engineering embryonic stem cells. Let me conclude the session with Einstein's quote, "Imagination is the highest form of research." Thank you.